We've reached a point in science where it's possible to find out all the letters in your DNA sequence, and that's pretty cool. But if you've ever looked at a DNA sequence, you might have noticed it isn't exactly English. We can find out all the A's, the G's, the T's, and the C's in a human genome, but it's a completely different language than the one you likely learned in grade school. And how can you tell which of these letters made your eyes blue and which ones gave you those big ears? Well, fortunately, your cells can read your genome language just fine, though it takes a few steps. Your DNA tells your cells what proteins to make. Remember that it does this through the central dogma, where your DNA is first transcribed into RNA. DNA is a huge manual of instructions, so transcription is like writing down one page of DNA instructions at a time. There are 3 types of RNA made during transcription: messenger RNA, or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. In the second step of the central dogma, mRNA is translated into protein during translation. You'll remember that proteins are built from several different amino acids in a specific order. So after that refresher, it's time to introduce you to the smarty pants of the cell who is able to speak this DNA language. This would be the ribosome, the cellular structure that performs translation or protein synthesis. Ribosomes are both language translators and efficient assembly workers. Putting together any sort of protein is like putting together a piece of furniture that has 3 boxes of more than 100 parts. And the instructions are in a foreign language, with no pictures. We've all tried to do it, and it's not pretty. But for ribosomes, this is a cinch. They read an mRNA sequence, translate it, and use tools to build it into a protein. This protein is then used by the cell. Specifically, ribosomes use tRNA as tools. This helps bring the correct amino acids to the building protein. Ribosomes are composed of rRNA and proteins. They exist free in the cytoplasm until they receive an mRNA message to translate. When they receive it, they waste no time in cracking the code. Ribosomes can be either free ribosomes in the cytoplasm of the cell, or they may find that the mRNA they are translating has a specific destination. In this case, some ribosomes will attach themselves to another cellular structure, and this is called the endoplasmic reticulum. Here, these are called the bound ribosomes. Proteins produced by bound ribosomes are usually destined for some kind of specific structure, or they're meant to exit